Hi, this is Dr. J. Bailey, and in this video, we're going to focus on topic 7.2, which is all about transcription and gene expression. So I went back and looked for most common questions when it comes to topic 7.2, and that means I'm going to focus only on the short and long answers related to this. Now, whenever you start reading these long answers, please look at the first word, the command word, because that's very important. It helps figure out what we are asking for. And the next thing we look is also for the points. If it's 1 or 2 points, that means go straight to the point and be very direct. If it's 5 to 6 or even more, up to 8, that means go for everything that is general to very detailed. So that out of the way, let's start looking at the first question. It says right here that to compare the process of DNA replication and transcription. So when you compare, in the old curriculum, it would have been just looking the differences between them. The new curriculum, compare would be known as similar. But in this case, since I'm looking in the past IB exams, you're probably going to see in the mark scheme both similarity as well as contrast. So with that in mind, I'm going to put here DNA replication. And here I'm going to put transcription. That is something very common is where they try to compare transcription with translation or replication with transcription. This is very common. So what I'm going to do is here as put in the middle as in what they have in similar. Now, if you know both similar, they're both happening in the nucleus, obviously. They both are using nucleotides, right? And they're both using DNA as a template. These are very common things that is happening between them. Now, DNA replication is actually make a copy of the DNA, right? So you're making 2 copies of DNA, and transcription, you're making 1 mRNA messenger. DNA replication, in this case, you're using different types of enzymes. In this case, we will say that we're using DNA polymerase. We can say the 3 or the 1. We can also talk about the other enzymes such as gyrase and the helicase, and we can list them all here. In this case, we're using the RNA polymerase. Now in this one, we're doing the entire DNA, right? Just the entire thing. Here, we're doing just one section of the DNA, which is basically we're doing one gene, right? What else can we say about transcription and translation? Transcription, after transcription, you have the splicing of the introns, right? And replication, you don't have any kind of splicing. That means you're taking the entire DNA. Here, obviously, you're going to be using the A, T, C, and G. However, in transcription, instead of the T, you're going to be using the A, U, C, and G. And I think this is enough going point. So let's look at the mark scheme and see what they have. First, both involve unwinding the helix. Oh, that's something common that I should have here. That it does have the unwind of the helix. But you got to be careful because here the helicase does this, but in transcription, it is the RNA polymerase. What else we have here? Both involve separating 2 strands. So these are just common, obvious things. Both involve breaking the hydrogen bonds. That's an interesting one. I didn't think about that one. Both involve complementary base pairing. That is true. That means they're all working with the A, one is working with the A, T, U, the other one is working with A with U, one is working with C and G, and here is you can see G. So that's what I mean by complementary base pairing. Both involve C and G pairing. That is true. They both work from a 5 to 3 direction. Yes, they're all going to build up from a 5 to 3. Replication, now they both involve leaking the nucleotides, and replication with DNA nucleotides, transcription is with the RNA nucleotides. Now here, I guess this point on, they start to see the differences. Details of ribose and deoxyribose is the difference. That is true. Why? DNA, all the sugar is known as deoxyribose, and in RNA, all the sugars will be ribose. Adenine will pair with the uracil instead of the thymine. And only one strand copy, not both. When it comes to mRNA, of course. No ligase or Okazaki fragments in transcription. Do we have any more? Yes, and of course DNA or RNA polymerase, depends on which one we're talking about. Both require a start signal. That is true. See, for the transcription, usually it will be the promoter. And for the DNA replication, it would have been, in this case, the signal could be anything from mitosis, basically telling it it's time to go through the S phase, and the S phase will start the replication. Both signals is different for each other, and transcription has only one starting point, which is definitely the start codon, if you remember that. The start codon. Well, when it comes to DNA replication, you can have here a DNA, and you can have a helicase starting here, another helicase starting here, and another helicase. So you're opening in different directions. That's what it means, at different multiple points for DNA replication, which is has multiple points here. The replication gives 2 DNA molecules, while transcription gives 1 RNA molecule. And that's pretty much it. So I did target some of these, but now that I'm exposed to the mark scheme, I can have better ideas. None of these ideas are like, oh, I never knew them. I never thought about them, but I know them. So now that I'm exposed, I have better knowledge. And that's why it's very important to go through these mark schemes of past papers. So what we have here is that up to here, all of these were considered as similar. And down here was all their differences. Now, usually if you see a list so huge as this, obviously this would have been an 8-point assay for sure. So with that, I probably will work on a 4 to 4 or a 3 to a 5 or vice versa. And that way I can get all the points. But you have to mention at least some similar and some differences. Now, here is the helicase, which makes sense. Helicase opening, breaking the hydrogen bonds, opening the 2 strands, obviously. Now, the most common thing is both of them has the complex bearing. However, this is just C and G, doesn't have the EG. Obviously, they both work in a 5-3 direction, and they both involve linking nucleotides, right? You're just building nucleotides after nucleotides. Then it comes the differences. One is involving the DNA nucleotides, but this one is RNA nucleotides. One is involving the ribose sugar, the other one is the deoxyribose. And here you're using adenine with uracil, while this one we're talking about using thymine. So basically this is happening in transcription, and this is happening in DNA replication. And then here, one strand copied, not both, for the mRNA. But for the DNA, you can say there is no ligase and Okazaki fragments transcription, but it does have a DNA replication. One has DNA polymerase, the other one has the RNA polymerase. I'll know that this one is for DNA replication, this is for transcription. Both require start signal, but that they are different from each other. So this should be usually as 1. The transcription has 1 point, the DNA has many points, and the replication is of 2 molecules, while transcription gives only 1. So if I was going to write here, it really depends on you. Pick 4 for each. If I was you, even go 5, just to be on the safe side. So if I'm erasing here, what I would do is, what are the most common things that I for sure will remember? Like me, if I would pretend the students, I would talk about the helicase, the helix, which is DNA opening up the 2 strands with the hydrogen bonds, the complementary base pairing, and the 5-3 and the pairing. These are very easy for me to say, even bonding bunch of nucleotides. So similar is very easy. Then for what are the things that I can't remember the difference? Oh, I can say one is using DNA, the other one is RNA, one has ribosome, one has deoxyribose, one is using uracil, one is with the thymine, one has DNA polymerase, the other one has RNA. What else I can say? Transcription has 1 point, DNA has many points, replication gives 2 molecules, while transcription only gives mRNA. Like, I now have gone and read twice this mark scheme, so that means I have a better knowledge. Why not write it down? Now you can pause this video and write down your top 5 for each, or top 4, and this way every time the mark scheme, the question comes to compare, you already have these in your mind, so your chances of getting full point is higher. Now the next question says, explain transcription and explain the process of transcription in prokaryotics. These are asking the same thing. So we're going to look at the mark scheme and see how common they are with each other. But when we say about transcription, we're definitely talking going from a DNA to be transcribed to an mRNA. That's where you have to talk about the entire process. You talk about the RNA polymerase. So we can also talk about the promoter, because that's where the RNA polymerase comes in. It touches the promoter, and after it touches the promoter, it starts to read the gene coding, right? And then once you start reading, what are you doing? You're adding nucleotides, but we, instead of using the T, we're using the U. And then there's a terminator, which that means when you finish the whole transcription, and that's when you have created an RNA that is released. This is basically, and then we can say that we are reading the antisense strand. So the mRNA will look like what? Looks like the sense DNA, which actually codes for the actual protein, right? What else can I think about this? I can talk about how there is transcription factors, right? As well as regulatory genes and how they control the transcription, which I can even add to this the methylation, which it controls. More methylation, less transcription, less methylation, more transcription. So these are the things I can write. Now let's see here. We have the two mark schemes, and we're going to see what they have in common, what they have in difference. So looking at the left side, says here synthesis of mRNA. And here, do we have anything saying about synthesis of mRNA? No, it does not mention any way about that. The next thing it says RNA nucleotides linked together to form a strain. Do we have anywhere here where we are saying you adding the nucleotides together? Nope. Here, RNA strands assembled on DNA template, antisense strand. Do we have anywhere we're talking about the antisense strand? Yes, we do. We have the antisense strand here, so I can mention about the antisense strand with the strands right here. Now, look at this. It says the RNA polymerase covalently binds the NAP, which is basically the nucleoside phosphate, which are the free nucleotides, together in the reaction that involves the release of two phosphates to gain energy, right? So this is also kind of talking about how the RNA nucleotides are linked together to form RNA. Now, what else? RNA carries out all the transcription and links the nucleotides. That also falls with this. Now we have the uncoiling and separation DNA strands. Do we have anywhere here? Yes, we do. Unwinding of the separation of these strands go with the uncoiling. Here it says 5' nucleotides linked to the 3'. It does have it, kind of indirectly saying here when synthesized from a 5 to 3 direction until it reaches the terminator. Does it mention about base pairings? Yes, it does. Uracil with adenine, cytosine with guanine. So we can say the base pairing together, which is uracil instead of thymine. Now start at RNA binds to the promoter. Do we have here? Yes, we do. See, it starts with the promoter, so we can say that this starts with the promoter. Regulated by transcription factors. Do we have that? We don't have that. So on the other side, what do we have in addition? We mentioned what a promoter is, which is starts the transcription. We coding sequence, which is a sequence that is actually transcribed. May contain introns, of course, and terminator is a sequence that serves to terminate transcription. And here is also mentioned about the terminator, the RNA polymerase, and basically the RNA gets to the new form strand and both detach from the antisense template, and DNA is rewind. Many RNA polymerase enzymes can transcribe DNA sequence, sequentially producing a large number of transcripts. Here it says regulate transcription factors binding DNA proteins and nucleosomes. So you can see there's some differences between them, right? The green parts are the things that they don't have in common. Also, you have here the post-transcription modification is necessary for eukaryotics. So here you adding some information because it's talking about the eukaryotics, right? Like the introns are happening in the eukaryotics. Remember, there's no introns in the probe because, again, the introns is only for the eukaryotics. And then here you have another thing, synthesis of mRNA, which it doesn't even mention, and the strand assembled DNA. So you can see that the blue is what they have mostly in common and the greens they don't. So what do you do with these kind of mark schemes? And here on this one, it actually allows you to draw it. So first of all, what do? Join the blue ones, make them into one. That means you definitely talk about RNAs getting to the promoter, then the RNAs opening the DNA strands into two sections, and then start building from a 5 to 3 the mRNA. And that means we're doing this by complementary base pairings, which is A with U and G with C. And it keeps going until we reach the terminator, and that's where it stops the synthesis and you have formed the mRNA and everything disassembles. That's the main thing. What you can do with that, mention that you're going from DNA to RNA, mention this area here, what is a promoter, what is a coding sequence, and then mention here how, again, there's a post-transcription modification, which means you change in the mRNA, or talk about here the regulating transcription with the nucleosomes. So these guys here, these two, there's something that you can add after you have talked about the transcription on both sides. So here is just showing two different mark schemes, and you see they're not exactly the same, but by you combining and making one mark scheme, then you will never get any points off on this mark scheme. Now here we have two questions. Nucleosomes help regulate transcription in eukaryotics. State the components of nucleosomes. That state will be saying a word, so this case can be histone and DNA, because that's what nucleosome is made. And you can go more specific, saying 8 histones for that one DNA. Now state one role of nucleosomes, and that is definitely to regulate the transcription, okay, or even to coil the chromosomes. So let's see what we have here. DNA and histone help supercoil, like I said, or help regulate transcription. So that you know right here the two functions of nucleosomes, and you know here the composition of nucleosomes. You know this, you already kind of start at base on nucleosome. Now, here says nucleosomes help regulate transcription in eukaryotics. State a chemical modification of the nucleosomes that could impact the gene expression. That is obviously methylation or acetylation. Now, what's the difference between methylation? This will actually cause the coiling, and that means it reduces transcription. However, if you have acetylation, this will loosen the coil, and this will increase transcription. So it's very important that you know these two differences, because it does come a lot when it comes to section 7.2. So when it says state, obviously, and this means 1 point. So if I mention methylation, I already got the point, or if I mention acetylation, I already got the points. Of course, there's other things like modification of the nucleosomes, the N-terminal, the phosphorylation, but I would focus on these two because these are the most common answers that I see when it comes to modifying the nucleosome. So remember, methylation, acetylation, they modify the nucleosome. Next question. It says, explain the control of gene expression in eukaryotics. So when we talk about gene expression, we can talk about making proteins, right? So we can say about the codon, how the codon basically expressed to an amino acid, right? And the codons are found in the mRNA, and when they are going through translation, they eventually turn into a protein, right? So this is gene expression. So one of the first thing we do is going from a DNA to transcribe DNA into an mRNA. Here you're using nucleotides, that's one. Another way to control transcription is we can also talk about the promoter, right? Because the promoter is the one who says, where do we start with the gene? We can talk about the terminator, because that's where you stop basically transcribing the gene, right? We can talk about nucleosomes and how they basically control transcription. And we can talk about the methylation, right, which can also be inherited. So you can be born with some of it or not. And this one, basically, like I said, it slows down transcription, while acetylation does the opposite. Acetylation, it basically speeds up the transcription. So let's go ahead and look at the mark scheme. So first it says mRNA conveys genetic information from DNA to the ribosome where you keep making peptides. So I did mention this. Gene expression requires production of specific mRNA. Like I said, you had to go and create the mRNA. Most genes are turned on off being transcribed as one-time regulated. Some genes are only expressed at certain times. That is true. So it depends on what time it can be. It can be when you're a baby or when you're adult. So this is a very important thing. They are expressed in a certain time. Another one is that some genes are only expressed in certain cells. That's another place, certain cells. So it's basically talk about cell differentiation that involves. So the time and the place makes a huge difference in which gene you're going to express. It's basically like saying that you have this one gene, okay, and depends on environment A, it will produce protein A. If it's environment B, it will produce protein B. If it's in environment C, you produce protein C. But it's exactly the same gene. But you find that gene in every single cell of your body, but depending where it is, if it's in the liver, it might do something else. If it's in the heart, it might be expressing something else. So the location can affect the gene as well as when you're going to do it. Transcription factors, proteins can increase or decrease transcription. Like I said, transcription factors is basically regulating like silencers, activators. Hormones, a chemical environment can affect the gene. That is true. Sometimes when you have more of a hormone, you can produce a certain type of thing. So an example of a hormone could be insulin. Insulin, the more insulin you have of that, the more you're going to have the beta cells, or in this case, the beta islets found in the pancreas. What are they going to do? They're going to produce more insulin. Or if you have a high spike of glucose, right, this also is going to make the B cells produce more insulin. So depending on the environment, if you have a lot of the environment sending a message to you, the more you're going to produce. So example cell environment, yep, you can see there's the insulin, auxin. So these are examples of how the hormone will actually influence. Transcription factors may prevent or enhance the binding of the RNA polymerase. This is basically we're talking about here. We have the promoter, and then here you have the gene sequence. This is where the RNA will actually, the enzyme RNA polymerase will bind here and start creating the mRNA. But right behind the promoter, we have control elements such as the distal and the proximal elements, and you have the transcription factor binding here, and we also have regulatory genes coming here. And what do these do? When they bind here, they will tell them to go forward or not. So we usually say they are silencers, or we can say they are enhancers. What does it mean? Silencers, they will stop transcription. Enhancers, they will enforce transcription more. But they usually bind here on the distal, and they will tell transcriptive factors to bind on the proximal. So we can talk about nucleosomes limit the transcription factor or regulate the gene expression by activating or silencing the genes. As I mentioned, it can be DNA methylations or appear to control the gene expression. Remember, the more methylated, the more silence, the more acetylation, the more it's activated. Some DNA methylation parents are inherited. Like I told you, the DNA methylation can be passed from your mom to the child. And introns may contain positive or negative gene regulators. Gene expression can be regulated by post-transcriptional modification, splicing. Yes, depending on which introns you cut, you're going to get different types of proteins. So all of these are examples of gene expressions that you must know. So I'm going to go over this again to reinforce the information. So when I talk about gene expression, first talk about mRNA. What does that mean? The production of mRNA, it's so important to do transcription translation. Next, when do I turn off those genes? Usually the time they do it, as well as the location of doing it. Then, who can cause them to increase or decrease this entire process? The transcription factors, right? They can increase, decrease, as well as the methyl and acetylations. These guys can increase or decrease the transcription. Now, when we talk about the environment, we also have hormones and chemicals that can affect. I give an example like insulin. So, and then I go again here, transcription factors may prevent or enhance the binding of the RNA. That means they can start the transcription or we stop. We also know the nucleosomes can also regulate the transcription, right? The gene expression. Then we can talk about the splicing that happens with the post-transcription modification. I mean, since we talked about the whole production here of mRNA, why not talk about also the editing of the mRNA and how can you make different types of protein depending on which introns or exons you're keeping or taking out. So as a total here, talk about mRNA, talk about nucleosomes, talk about transcription factors, talk about having some genes being exposed to different timing and locations. You have talked about all types of ways of controlling the gene expression. So here I'm going to put like a cheat sheet for you quickly. So the first thing is talk about what? Talk about gene expression, right? So when you talk about gene expression, one, talk about mRNA production, right? How do you produce it? Number two, talk about how mRNA is spliced, right? And this causes to have what? Different proteins. Then three, talk about gene location and time. That makes a huge difference in how they're going to be expressed. Number four, talk about transcription factors. Will they increase or decrease transcription? Since we talk about transcription factors, we can also talk about nucleosomes. What do? They regulate transcription. And since we're talking about nucleosomes, we can also talk about methylation as well as acetylation. So this can speed up or slow down transcription translation. So right here, I already have enough of a cheat sheet. And now you have this, you can pause and write these down. But of course, these are just an outline. Of course, you have to elaborate everything about how mRNA is produced, how it's spliced by taking the introns out. Then talk about gene location, might turn off or turn on at a time. The transcription factor can increase or decrease transcription. Nucleosomes can regulate transcription, which that means including methylation, acetylation. And of course, I forgot 7.1, which is the environment can affect the gene, right? And this case can be a chemical messenger or a hormone. And I can give you as an example the insulin hormone. So here I have a very strong outline base talking about all the possible things of gene expression. And if you have these 7 points written, and of course you elaborate on them, you got a very perfect mark scheme to talk about gene expression. So make sure to jot this down and make sure to know each one of these. Now in my lecture, I have talked about them. So if you get a little bit of like, if you mention 2 points or 2 informations for each one of these guys, you have a very solid answer for gene expression. Now here says insulin is produced in B cells of pancreas and not in other cells of the human body. Explain how differentiation of cells and regulation of gene expression allow proteins such as insulin to be produced only in certain types of body. Now the fact that it says can only be produced in one area, not, that means its location. So that means you can have here a cell, let's say 3 cells. This cell is found in the liver, this cell is found in the pancreas, and let's say this cell is found in the kidney. Now, all of them have the gene, right? The exact same gene. The only difference is in here in the pancreas, this gene is turned on. And this gene, what does this gene do? It actually produces the beta cells. And what does the beta cells do? It actually produces insulin. So that means only the location in the pancreas will the gene be turned on and produce the B cells, which eventually will create insulin. Now, turning the gene on and turning it into a B cell, this is known as differentiation, right? So you're basically turning a cell into a specific job, and the job of the B cell is to produce insulin. So that's differentiation. You give him a specific job. Now, in this process, that will be the part about differentiation. Now, when it comes to regulation of the gene expression, this is when we're talking about transcription. And when we talk about transcription, that means we want to create more insulin. If we want to create more insulin, first we have to find the messenger. We have to create the mRNA by finding the gene in the DNA that creates insulin. So by knowing the gene on the DNA that creates insulin, you convert them into mRNA to transcription, and then the mRNA is read, and from there we go to translation, in which translation will create the insulin. Now, to trigger the gene or DNA all of this going on, obviously we need to have glucose in the blood, because glucose in the blood will lead the gene to say, hey, we got glucose in the blood, so we have to create more what? Insulin, because you want to get that glucose into the cell. So by having more insulin, more glucose gets to the cell. So by glucose being in the blood will trigger the gene to be what? Turned on. Now, this will only happen at the beta cells of the pancreas. And then if it's turned on, then we're going to do transcription right here, which will create the mRNA, which is the message to make insulin. Then we go in the ribosome, do translation, and voila, we have produced the insulin. And then the insulin will go and put the glucose in your cell. So we have showed two things here. Cell differentiation, which means the gene was only turned on in the location of the pancreas. And regulation of gene, we means if we need to make more insulin, we're going to do transcription and translation, but we have to turn on the gene first. And of course, from there, we create insulin to build it up. So first, insulin production is determined by the gene. That's the whole process of saying you have to activate the gene to make insulin. Second, the gene for insulin found in all cells, but only activated in the pancreas. Like I told you, you can find the gene in all cells, but it's only activated at the pancreas. Stem cells differentiate in specialized cells. So in this case, we're saying that the cell became into a beta cells, and only the beta cells produces the insulin. Next, during differentiation, some genes are turned on and others off. That's what we showed here. When I showed the liver, the kidney, and the pancreas, only the pancreas turned on its gene. And then the insulin hormone regulates the amount of glucose. I have mentioned what insulin does. Then the pancreatic cells have sensors that detect glucose levels. So right there, I was mentioning how you have so much glucose in the blood. When you have so much glucose in the blood, now it's time to do what? It's time for the pancreatic B cells to produce what we know as the insulin. Then increased glucose will increase transcription. Why? Because again, if you want to make insulin, you have to make the mRNA. That means you have to go to the DNA, which has the gene, and find the message or build up the message in order to produce insulin. Then the site of transcription of insulin is in the pancreatic B cells, obviously because we know the B cells is where you produce the insulin. Now, gene transcription is regulated by proteins that bind to specific base in DNA, such as can be the enhancers, silencers, promoters, and proximal elements. So this is just an additional information saying that if you want to start the transcription of a gene, you need to have the promoter, you have to have the enhancers, or the silencers, or even transcription factors. Regulatory sequence proteins are specific to the gene they regulate. Insulin-regulated proteins are only found in the pancreatic B cells. Makes sense here. The regulatory proteins, they are the ones who say, yeah, go ahead, make more mRNAs for insulin or not. And DNA methylation usually happens in gene expression. So since we are talking about gene expression, every time you read the information in gene expression, why not talk about methylation, how methylation can actually stop the transcription, while acetylation can actually enhance or promote the transcription. So whenever you see the word regulation gene expression, don't forget to talk about methylation and acetylation, because these two always come in the mark schemes of regulating gene expression. I guarantee you. So right here, we have all the information. Talk about the differentiation, which that means the genes are located in every cell, but only one of them activates it. To activate it, of course, you have to have a certain kind of trigger. In this case, was glucose in the blood. And then that causes the pancreatic to differentiate their cells into B cells. And then the B cells will produce insulin. But to produce insulin, you have to do transcription and translation. Now, when you're doing transcription, you have to regulate it. So you have to activate it through acetylation. You can activate it through transcription factors, promoters, and so forth. So this is a very strong example of gene regulation using insulin as an example. And that's pretty much it for 7.2. Here are the future possible questions that can come out for 7.2. They have been answered in my lecture video. So if you go there, you have everything. Now, I do feel like I want to come back here and put this in a better drawing so we can really master, because this is a perfect example of gene expression. So what I'm going to do is be nice here and put this in a drawing so you can really master the insulin. So with that, I'm going to start here by mentioning here again, imagine you have 3 cells, right? One is found in the liver, one is found in the pancreas, and the other one is found in the kidney. So you can see these are three different what? Location. Now, all of them, like I said, they all have an exact same gene. So this is a gene. In the DNA, they all are exactly the same. However, only in the pancreas was this gene turned on. So you can see that the liver, it was not turned on, and the kidney was not turned on. So the gene was turned on here and turned a cell inside the pancreas to become who? To become a beta cell. So this became differentiation, right? Because you're giving a function. And what does the B cell do? It produces insulin. So this is known as differentiation. A cell, a stem cell is turned into a B cell, right? I should put here stem cell so you can see into a B cell. This is only because the gene was turned on and that gene caused information to say, hey, stem cell, become a B cell, a beta cell. And B cell has a specific job. So that means B cell can only be found in the pancreas. So this is the only one that has been turned on. Then, now we know that the B cell has to produce the insulin. Now, let's just say you have insulin here. Oops. What does the insulin do? The insulin, its job is to put glucose inside the cell. Why? Because you want the cell to use glucose to what? To get energy. That's cell respiration, when you're breaking down glucose to get energy. Now, let's just say you have a lot of glucose in your blood. So I'm going to put here arrow saying a lot of glucose in your blood. This will cause what? A response. That response means create more insulin. Because if you have more insulin here, then you can put the glucose into the cell. Now, if you want to create more insulin, what do you have to do? You have to go all the way back to the beta cells and say, hey, beta cell, I need you to do insulin. Now, remember, the insulin is a protein hormone. So if you want to create the protein, what do you need to do? You need to go through transcription and then eventually through translation. So if you want to go to transcription, what do you have to do? You have to basically find the DNA, the gene, and then transcribe it into mRNA, right? And to do that, you have to see the regulation transcription. So you have to activate transcription. Now, who regulates, who controls transcription? The nucleosomes. They can control the transcription through what? To the methylation or acetylation. Now, what is the difference? Methylation will decrease transcription, acetylation will increase. So acetylation will produce more insulin and methylation will produce less insulin. Another thing is we can have is regulatory genes or proteins. They're the ones who say, go ahead and cause transcription. That will be the answer. So go ahead, stop it, which is called silencers. So these all tell you exactly if the B cell is going to go forward or not. So this was just a drawing to get you an idea how we're regulating the genes. But we have to know that when we are doing regulation of the gene expression, we are also doing cell differentiation, because everything starts with reading the gene. So I hope this helps a little bit more understanding how insulin is produced. And that's pretty much it for 7.2. Thank you.